start in the interest of time. In particular, since there will be a coffee break or a special coffee break, I guess, after this presentation. So I'm not sure what the special coffee break is going to be. Does anybody know what's the special special machine? Okay, so we'll have great coffee, I guess. So thanks for trying to. I'll try to not be too long in my presentation, so we can all enjoy this coffee. Okay, so what I'm going to present today are computational methods for the analysis of survival time data sets. In particular, when you have analyzed the mutations genome-wide in a large number of cancer patients. And these settings makes the analysis of survival data quite different compared to the standard analysis of survival data. So we're going to see what are some of the computational challenges that I've worked on in the last few years. So just as a quick recap for the ones that were not here during my talk or others' talk, nowadays we can measure mutations in a large number of cancer patients. And in particular, what we are interested in and what we will be looking at are somatic mutations. So changes in the DNA that appear after or during the lifetime of an individual. Okay, so just to give some concrete numbers, there are public data sets that are collecting somatic mutations for a large number of cancer patients. Like, for example, the International Cancer Genome Consortium. If you go to their data portal, you can download the mutations for more than 17,000 donors, and they are donors from more than 20 different cancer types. So these are all the somatic mutations that we can collect publicly. And just to give an idea of the numbers, in a typical patient, in a tumor, what we measure are between hundreds up to tens of thousands of somatic mutations. And maybe 10 of these are thought to be related to the disease. Yes?
When you say somatic mutations, you mean the difference between the tumor to a healthy tissue or the difference between the tumor to the reference genome?
That's a great question. So it's the difference of the tumor to the healthy tissue. Or usually it's the difference between the tumor sample, what you measure in the tumor sample, and the blood, for example, that is used as the standard normal tissue for the patient.
Do you always have both in your sample, like in BCGA, or sometimes you just have the tumor genome?
For the data types and the analysis that I'm going to present, we always have both. So you can get the data that are the somatic mutations, and they are essentially the difference between what you see in the tumor and what you see in the normal. So you take out the germline mutations in the patient. Okay, so these are the type of mutations we will focus on. And I guess on Thursday I presented, a class of approaches to solve the problem finding mutations associated with cancer. So how can we identify maybe these 10 driver mutations that should appear more or less in the patients? And I presented single gene approaches and other approaches, network approaches, and approaches based on combinatorial patterns of mutations. But what I want to present today is something different that doesn't focus only on the mutation data, but uses also a different data type that is available for at least some of these large cancer studies. And this data type is in particular survival time. So for some of the large cancer studies, we also have clinical information, and in particular the survival time of each patient, and we can think of it as just a real number. Okay, and so if you have now measured somatic mutations genome-wide or exome-wide, so you have the mutations in all the possible genes, in all the patients, and you know the survival time of the individuals, a natural question to ask is, what are the mutations that are associated with survival? Okay, so what I mean with this is, can we find a mutation or maybe a gene such that if I partition all the patients for which I've measured mutations into two groups, the patients without a mutation in the gene and the patients with at least one mutation in the gene, I observe a different survival for these two groups of patients. Okay, and of course this has important implications for therapeutic or for clinical decisions. Yes?
How do you define survival time? Like, is it how long the patient lives?
So it's the time to an event in general, and the event could be the death of the patient, there could be a recurrence. In general, the survival analysis is the analysis you do for when you consider time to an event. Okay, so for most of the data in TCGA is the time to death for the patient, but more in general, you could think of survival time as time to an event.
So all the samples that we do of patients who had already died?
That's a great question. So what you're asking is, do I actually have, oops, I gave away the answer, I guess, on the next slide, but that's a great question in the sense that what you're asking is, do I really have the survival time for all the patients? And the answer is no, in the sense that actually, when I talk about survival time, I'm talking about two pieces of information. Okay? So the survival information that we have is the time to the event and what's called the censoring variable. That is just a binary variable that is, let's say, 1 if actually we observe the event at the given time for the patient, or 0 if all I have for the patient, what I've measured, is just a lower bound to the real event. So for example, the patient is, if we're talking about death, patient is still alive, or the patient has left the study at some point and I don't know what's the status. Okay? Yeah?
So do you just use the variance, or do you also incorporate somehow stage?
That's another great question. At this point, we are only looking at variance. Later on, I'm going to say something about how to incorporate stage maybe, but in the approaches I'm going to present, we're going to focus mostly on variance. Okay? Yes?
For these samples, do you know if they have gone through big treatments, are they having all similar treatments, or there might be difference?
It's another great question. So for some data sets, we know the treatment that they received, and then you can control for this variable. Yes, but only for some data sets, not for all of them. Okay? And then it depends also, if the data has been annotated well or not, but that's a different issue. Yeah?
Timing from the point that you took the sample?
Yeah, that's the time from the sample was taken up to the event or to the lab observation. Okay? So I guess I'm already behind in my presentation, so let's see how far I can get. But I'm going to just speed up a lot at the end so you guys don't understand anything, but you will keep having tons of questions. Okay, so let's say that, so if I want to identify the mutations associated with survival, and this is the data that I have for each patient, I have mutations, the survival time, and the censoring observation. What I can do if I focus on one specific gene is partition the patients into two groups. So just assign a group label to the patient. The group label will be 0 if the patient doesn't have a mutation in the gene, is 1 if the patient does have the mutation in the gene. This is essentially the partitioning of the patients into two groups that I discussed earlier. And then I can use standard tools from survival analysis to estimate the survival probability for these two groups. It could be, for example, the curve in blue for the blue group and the curve in red for the red group. These are Kaplan-Meier plots, if you have ever seen them. And then I want to understand if the observed difference in survival for the two groups is significant or not. So what I can do is I can use a statistical test that will tell me what's the probability that I observe a difference in survival, at least as extreme as the one that I see in my data, assuming that there was actually no difference in survival for the two populations. Okay? So just abstracting a bit, the output of the statistical test is going to be a p-value that is small if there is an association of the mutation with the survival. Okay? Okay, so now if I'm able to do this analysis for one specific gene, I can do it for each gene in turn. So I can do it for the first gene, partition into groups, estimate the survival probabilities. I do a statistical test, I get the p-value for the first gene. I can do it for the second gene. So now I partition the patients according to the mutation status of the second gene. And the partition in general will be different from the partition that I had up here. And I get a p-value, and I do it for the third gene, and so on and so forth, for all the genes that I have mutated in my data set. And then in the end, I can, for example, rank the genes by p-value to see the one that has the highest association with survival, or I can do a multiple hypothesis correction to find the ones that are significantly associated with survival. Okay? So that's quite easy, but we can see that what's important is this statistical test that gives us a p-value. So luckily, the field of survival analysis has studied this problem for at least 60 years, I guess now. And so there are methods to perform such analysis, for example, the log-rank test. So the log-rank test is a test that was originally designed for clinical trials and is a non-parametric test that assesses the difference in survival between two populations. And it is non-parametric in the sense that it doesn't assume any form for the survival distribution. Okay? So to give you some more information about the log-rank test, we can think of it as a function of the data, where once I have sorted my patients by increasing survival time, then I have two vectors, C that describes the censoring information for the patients, and the vector X that describes the group label for the patients. Okay, so the censoring is 1 if I have observed the event at this time and 0 otherwise, and X is 0 if the sample that has the event at this time is in group 0, and it is 1 if the sample is in group 1. Okay? So essentially, these 0 and 1 split the patients into two groups, the blue and the red, that we've seen before. And then what I compute with the log-rank test is a statistic that depends only on these two vectors now, on C and X. Okay? And in particular, the statistic is going to be a function of X and C divided by the standard deviation of the statistic under the null hypothesis and the null distribution. And in fact, since we are considering one fixed data set, one cancer data set, we can assume that the censoring is fixed. Okay, it's going to be the same for all the genes that we look at. So actually, what we are going to care about is just this grouping given by the mutations in the gene. Okay, so we can really forget about the time and the sensory information, and all we care about is this group label once we fix the cancer data set and we have sorted the patient by increasing time. Okay, so we can essentially consider now this statistic as a weight, and we'll see later how we use this weight for the second part of the talk. Okay, now under the null hypothesis of no difference in survival between the population 0 and population 1, the population of patients with no mutation in the gene, the population of patients with the mutation in the gene, we can compute the p-value as the probability of observing a weight at least as large as the one that I see in my data under the null hypothesis. Yeah?
So just trying to understand this statistic.
Yep.
So what are the quantity, V and V?
Okay, so V is actually the test statistic and is just the sum across the different time points of, or the time points in which I have an event, of the difference between the observed number of events in one population minus the expected number of events for that population. So now, if I, once I have conditioned a particular time, I will have two groups of, let's say, n0 elements and 1 elements. And now if the survival distribution is the same for the two populations, the probability that I see an event in the group 0 is going to be n0 divided n0 plus n1. And then I take the difference between what I see and the expectation, and I sum across the different time points. Okay? And this is the standard deviation for that quantity. So it's a standardized statistic. Okay. Okay. So I started this work, I guess, in 2012. So we were just looking at the data and was, okay, great. After learning a bit about survival analysis, I said, I found, now I understand what's going on, and let me implement this just simple scheme that we have described for some real data. So I took data from the TCGA, glioblastoma multiforme. There were 268 patients, and 30% of these patients have a censored observation. So for only 70% of the patients, I have the actual measurement. And we consider the genes with frequency at least 1%, and we did the scheme that we described earlier. Okay? So I do the survival analysis for each gene in turn. I used R-SARF-diff to compute the p-value, and then I do my multiple hypothesis correction. At the end, the result is that I get 110 genes with mutations associated with survival after Bonferroni correction with alpha equal to 0.05. Yes?
And in these kind of analyses, do covariates like population structure, do those things matter?
We didn't do it here. You can do the same thing using the Cox proportional hazard model to to incorporate these other covariates, so population structures, age, sex, and different covariates. You would get the same results for what I'm describing now. Then later on, I'm going to focus on the test without these covariates. And then the interesting thing to do is to extend what I've done when you have these covariates to take into account. Right now, we do it only afterwards to correct for those, but to do that in the algorithms, that would be very interesting. Okay? Yep.
Did you normalize for the overall rate of mutations in patients? Like, you say that the gene could have many mutations, but it does not mean that it causes people to die. Maybe just people that die have higher mutation rate.
Yeah. So what you are, if I can maybe rephrase what you're saying is the number of mutations in a patient may be correlated with the survival time.
Right.
So that should be a confounder to take into account. Yeah, you can do that as well, and you get similar results. Okay. Oops.
You can also make a less conservative correction if you want.
Yep.
Right?
I can do less conservative corrections. I can use correction for false discovery rate. That's all great. But the main point that I want to give you here, and that I think I already showed on the slides a few times, is that then actually, when I look at the data, I realize that I had run this method on a random data set. Okay. So these are the results on random data. What do I mean by random data? Is that I was doing a lot of simulations, and I had generated in dataset in which the mutations are the same, so they're fixed. What I'm doing is I'm permuting the survival time in the censoring together for all the possible patients. At the end, I do all my analysis, and I get 110 genes with mutations associated with survival.
Again, the same number.
The same number. And these are all false discoveries. Okay? Then I do it again, and I get different numbers. I do it many times, I get different numbers. But every time I get a false discovery. But instead, since what I'm doing, I'm supposed to see a false discovery around 5% of the times. So what's going on? Okay, so first to understand that this was a problem, not just a bug in my code, took some time, and then there are many steps that I'm skipping just to keep the interest hopefully high. Okay, so what's the problem? Well, the problem is that actually we were using standard implementations of the log-rank test. Okay, so the standard implementation for the log-rank test are based on a very nice result that is an asymptotic approximation for the distribution of the log-rank statistic. So under some assumptions, the distribution of the log-rank statistic follows asymptotically a standard normal distribution. So it's a martingale, and then you can prove that it converges a normal distribution once you normalize it. And one of the assumptions that you make and that you need to have this result is that actually you have balanced populations. So if you remember, I said the log-rank test has been designed for clinical trials where you have many cases and many controls. Okay? But in this analysis of cancer mutations, when you look at somatic mutations, you split the groups into two based on the mutation status of a gene. You have a small sample with a small population with a mutation and a larger population without a mutation. So in this case, what happens is that if you look at the QQ plots of the p-values that you get under the null, they are actually not uniformly distributed. Okay? So we did simulations, and you see that the higher the imbalance, the higher is the deviation from the expectation, the expected uniform distribution for the p-value. Okay? So this is true when you have, let's say, 100 patients, and then we said, well, maybe you solve the problem if you take, let's say, 1,000 patients or more. And we did actually simulations up to 10,000 patients, and let's say 5% of the samples in one group and the remaining in the other group. And you still see this deviation from the uniform distribution. So you cannot really use this asymptotic distribution when you're doing a survival analysis for a gene that is quite rare compared to the entire distribution. Okay? So after understanding this, we said, okay, so let's try to solve this problem. Just to make it even more clear, the problem is that for very low p-values, this approximation based on the standard normal is not precise. So what you need to do is to compute these p-values from the exact distribution. Okay? So we said, well, okay, now it's an interesting computational problem. Probably it has been solved already, so let's see if it has been solved. It hadn't been solved, so we didn't find any efficient algorithm to compute these p-values from the exact distribution. And so now I'm going to tell you what is this exact distribution, just to give you an idea of what is the computational problem that we need to solve. And then I'm going to tell you a few things about the algorithm. Okay, so what's this distribution, this exact distribution? So let's say we have our group labels, X, and in general is a vector of n entries, and n1 of them are ones. So these are the mutations. And under the null hypothesis, or the null distribution of no difference survival, all possible n choose n1 locations of the n1 ones in the vector have the same probability. Okay? So computing the permutational p-value is just equivalent to ask how many of such vectors, of the n choose n1 vectors, have a statistic greater or equal than the one that I see in my data. So we call this the more extreme assignment counting problem, very long name for a simple problem. So given now these numbers, you want to compute the number of vectors that have a statistic larger than the logarithmic statistic that you see in the data. And this is actually answering, I guess, your question. This is the statistic that you observe. Okay, so as we said, there was no efficient algorithm to solve the problem. We tried to understand the computational complexity. Is it NP-hard? So in general, you cannot solve it easily. We weren't able to find, to prove that it is NP-hard. There are strong reasons to think that it is NP-hard because it is similar to other problems, but we don't have a proof to say it's really difficult or not. However, so the best we could do up to when we started the problem was to use an exponential algorithm that does just the exhaustive enumeration of the vectors to compute the p-value. So this takes time sent to the n1 that is not feasible for the data sets that we were looking at. Okay? So what we have done then is we have designed an algorithm that we called EXACT for exact long-rank test, not a very creative name, I guess. That is an efficient algorithm to approximate the exact p-value for the log-rank test while controlling the approximation. So what do I mean by while controlling the approximation? It means that actually the algorithm we give is a fully polynomial time approximation scheme for this counting problem. So let's see, how many of you have ever heard of FPTAs or fully polynomial time approximation schemes? Okay, more than usual, so I guess that was a good idea to put such name on the slide. So actually what we can prove is that for any fixed epsilon greater than 0, there is a polynomial time algorithm such that if p is the p-value that we want to compute, we can produce in output a value p star that is greater or equal than p, so it's a conservative estimate of the p-value, but it is less or equal than 1 plus epsilon p, where epsilon is given in input by the user. So we can control the approximation. Okay, and then the algorithm has a runtime that is not great, is quadratic in the number of samples, quadratic in the number of mutations, and then also times log n. Depends also on the approximation as well. So this is not great, and one open problem is how to improve and have a more efficient algorithm. And this is joint work with Eli Upfal, Ben Raphael, and Alexandra Papustaki, that were all at Brown University at the time. And we've implemented this algorithm and tested it on published data sets, on TCGA data, on simulated data sets. Okay? Now I'm going to just show you some results. So first, this is the comparison of the runtime of our algorithm. Here it's called FPTAs compared to the exhaustive. And so you can see that varying various parameters like n and 1 and epsilon, our algorithm is essentially always more efficient than the exhaustive one. So it's just a check to make sure that we didn't waste a lot of time to have a really nice algorithm that is not as efficient as we want it on real data. And then we try to collect some survival data from published papers to see actually if the p-values that we get with our tool are similar to what was published or not. So this was an interesting exercise because if you try to collect survival data from studies that have already been published, it's not so easy, at least my experience. So what we ended up doing was to reconstruct from the picture the vectors x and c, since that's all you need. You don't really need the exact time, right? You need the group label and the censoring information. From the picture, when you don't have too many samples, you can do it, okay? So that's why I'm not going to show you tons of results for this part. There are only few. The ones where some of us had the patience to stay there, reconstruct the vector, make sure we got the same results using RSRFD for the same tool. Okay, so for 2 studies in the Journal of Clinical Oncology in 2009, actually one study that has these 2 plots. So this is one of the figures they had, and they said, okay, the p-value here for the difference survival between these 2 populations is 0.012. It's interpreted as significant, and then we run our tool. And what we get is that the p-value is 0.17. So it's a 1 order of magnitude difference.
Maybe I missed something.
Yeah.
The values were for specific genes.
These ones are for specific genes.
So this is one underlying gene.
Yeah.
Is there a difference? Okay.
Yeah, so in these studies, they picked one specific gene, and they said, okay, let's compute the p-value for the difference survival between the 2 populations.
Correction issue, this is really just the underlying test statistic.
Yes, exactly. This is just the underlying test statistic. It's not the case of genome-wide survival studies, but we wanted to see what was happening in the some of the published data sets. Okay? Okay, so they got this p-value of 0.01. It was significant. Ours 0.17. We use the correction. This is up to, we use the 0.1, so it's 10% off at most from the true p-value. So it means that actually, if you look at it as significant or not, it's not significant. And more in general, I think there is 1 order of magnitude difference in the p-value from what's reported with the standard tool and what's reported with our tool. Here, instead, what's reported was a p-value of 10 to the minus 6, essentially, and with our method, we got 10 to the minus 3. So it's still significant if you want to use a threshold of significance, but there is a 3 orders of magnitude change. So maybe your interpretation for the results would change using different p-value. Yes?
When you said the difference is exact, you mean that like it's not relying on some permutation test, but it actually goes over all possible permutations?
No, sorry. What I mean is that here the p-value comes from the asymptotic distribution of the test statistic. Here it comes from the exact distribution. Okay. Still,
Computationally approximate.
that we computationally approximate.
But it's based on a permutation test, right?
Is not based on a permutation test, no. So our method is not based on a permutation test. If you did a permutation, so the null distribution is the permutational distribution, where you look at all possible permutations. When you want to compute the p-value from such a distribution, you either enumerate all of them or you do a permutation test, essentially a Monte Carlo method to estimate the p-value. But if the p-value is very small, then you need to do too many things. We have an algorithm that is completely different. I can tell you more later, but it's quite involved, and I would be happy to talk about that. That gives you actually an approximation that is controlled. So it's conservative. It's at most 1 plus epsilon multiplicative factor off from the true value.
And it's not based on some permutation, it's like analytical formula.
It's completely different. You start from the distribution, and you essentially, maybe I even have, well, let's see if I can say it in few words. Essentially what you do is you start from the first time point, you decide if there is a mutation or not, and you split your possibilities in 2. And every time you can have a mutation or not. So if you keep going with this process, at one point you have an exponential number of possibilities. And what we do is we collapse some of these possibilities in a controlled way, such that at the end you have this 1 plus epsilon factor off. Okay.
So we do not use the permutation test as the we are using PC values.
Not, so the standard, I look at the papers where I have to admit, in some papers, there was nothing reported about how the p-value was computed. Maybe just because it's standard, I'm not sure. And then for the ones that were reported, instead, they were reported tools that are based on the asymptotic approximation. There are some tools that are based on the permutation test. They are available already in R and the rest, but in those cases, it's not clear if you can really control the approximation. You can get a confidence interval, of course, but for most of the papers I've seen it, when they reported the tool, was based on the asymptotic approximation.
But granted the fact that you can control the degree of approximation, maybe it would be interesting to see on simulated data if you run, standard. So you have
Mhm.
asymptotic version of the test. You have a plain, no assumption permutation test with a certain number of random permutations, and then you have yours.
Yeah.
How do the straight permutations and your compare say, if you withstand the same computational burden, if one tends to do better than the other, or vice versa, you know,
Yeah,
how
yeah, yeah.
many random permutations do you need to run to actually be as accurate as you are, say,
Yeah.
or something like this.
So that depends really on the p-value that you would observe. Yes, right. So that's why we didn't do anything based on the permutation test. So we have the code available for this tool, and we also implement the permutation test, in the sense that if you want to screen 20,000 genes and you get a p-value of 0.05 with the permutation test with 10,000 permutations, you already know that you can discard the gene. You don't really care about computing it accurately, right? And then we have a two-phase where we first screen on permutation test, and then we use our tool for small p-values to be accurate on that. Okay. Okay, so for this other study, instead, the reported p-value was 10 to the minus 3. We got 10 to the minus 4, close, but you know it's even more significant that you would do. I want you to notice that here there are 129 samples, so we had to reconstruct manually sensor information group for all these samples. So that was a really interesting week, I guess, for the ones that did it.
This level of unevenness is standard in this type of studies.
Yeah.
2 and 14, 8 and 40.
Yeah, if you look at mutations, the fraction of patients with the mutation in one gene in cancer is almost always below, there are very few that are more than 10%, and vast majority of them are below 5%. Okay, so I spent, as I said, we started 2012, it took some time to understand, implement. Yes, exactly. That's, I didn't have glasses before starting this project. I'm kidding. Okay. So we implement these. Now we're very happy, it can be improved, but we have a tool. So now let's go back to our TCGA dataset, and let's now finally get genes with mutations associated with survival. So in 2 years, what was achieved was to replace rsurf-diff with exalt in here. So that's the result of 2 years of work. And at the end, we run everything, and we get no gene with significance associated with survival. Okay, so what's the conclusion? Well, first, it wasn't a great project to work on. It was a lot of fun. I've learned a lot. I think it was a very good project. Second, maybe I should just give up and say, okay, let's forget about this problem of finding mutations associated with survival. Or there is a third way, that is, well, maybe instead of looking at single genes, we should look at combinations of genes, right? The same way as we've seen on Thursday, if you were at my talk, that you cannot identify all driver genes by looking at them individually. Maybe what you need to do to find genes associated with survival is to look at combinations of genes. So, okay, so we solved in 2 years the easy problem. And now let's try to find a way to find sets of genes that are associated with survival. With the idea that, in general, the cell is a circuitry of interacting proteins and genes. And so, it may be that there is a set of interacting genes that perform a function, and maybe this is the function that is associated with survival. Okay, so second part of the talk will be quite quick. But let's try to find these subnetworks associated with survival. Okay, so let's first understand what we mean by saying that we want to find a subnetwork associated with survival. Let's say we have a subnetwork of 2 genes, gene 1 and gene 2. They are interacting in a given network. And what we can do is split the patients that we have into 2 groups, the ones that have no mutation in G1 and no mutation in G2, and the ones that have a mutation in at least one of the 2 genes. Okay, so this is the same, it's very similar to what we had done before with one gene. Now we do it with the OR of the mutations in G1 and G2. Yeah, the union of the 2. Okay, and then we can do exactly the same as we've done before. We have the survival distribution, we have a log rank test, we get a p-value. Okay, so this is if we give me already the subnetwork. But what if I want to find the subnetworks associated with survival? Well, so let's formalize the problem a little bit more. We have the mutation data, the survival information, survival time, and censoring, and we have, let's say, a large gene interaction network. And what we want to find are connected subgraphs such that, for example, one way to specify the problem is to give in input a value k and say that we want to find a subnetwork of k genes with the highest association with survival. Okay? So how can we solve this problem? Well, we need first to define a bit more what it means to have the highest association with survival. But this is easy since we have seen that the log rank statistic, we can really interpret it as a weight for the partition of the patients. So now given my subnetwork S, this defines group labels for the patients. And the log rank statistic for this partition, I can look at it as a weight, the weight of the set S of genes. So I can now specify completely my problem and say that given the graph G of n genes, the mutation matrix M, the survival information for the patients, I want to find the subnetwork, the connected subgraph of k genes that maximizes this weight. Okay, so this is the problem I want to solve. First result is that this problem is NP-hard, so in general there is no efficient algorithm to solve it for all possible instances. It's also NP-hard when your graph has only one hub, one high-degree node. That is the case for all protein-protein interaction networks that we're interested in. So it's, also for real cases, it seems to be difficult. We still want to solve it. So what we did is we relied on a technique that has been used a lot in bioinformatics to solve problems on graphs. That is the technique of color coding. How many of you have ever heard of color coding? I'm sure somebody over there has heard about color coding. Okay, not that many, so let me give you a one-slide introduction to color coding. So let's say that you have a graph, and you want to find a subnetwork that maximizes a function. And you want to find a subnetwork with k vertices that maximizes the function. So one approach to solve this problem is to random color the graph using k colors. So each vertex takes a random color among k. And now I have a colored graph, and the reason this is this is a good idea, is that now finding the highest scoring colorful subgraph in this graph is now easy. So what I mean is that if I want to find a subgraph of cardinality k that has vertices all of different colors, now this can be done with dynamic programming. And I'm not going to go into the details given the time. But this is quite efficient, and in particular is efficient is k is small compared to the total size of the network. In particular is of cardinality up to of the logarithm of the number of vertices. Yes?
Do you need to assume something about the function you are trying to maximize?
That's another great question. We're going to get there in two slides. Okay.
Because otherwise it seems anyway to go over all combinations with three different colors because you don't know anything about the function.
You need some assumption on the function. In particular, what you need is that your function is additive on the subsets. And that's actually the interesting twist for what we did. Okay, so given one coloring of the graph, I can find the highest coloring subgraph. It may not be the best subgraph in general in the original graph. So what do? I repeat this process enough times. And then if I repeat enough time, the highest scoring subgraph will be colorful at least once in one of my instances. So with high probability, I will find it. Okay. It's a very simple approach. It has been used a lot in bioinformatics for many different problems and with different twists. And I guess we may hear more tomorrow about one application in case control studies. So using this approach, we designed what we call the nomas for network of mutations associated with survival. And essentially, we combine the gene interaction network with mutation data survival, and we use color coding based algorithm to find the subnetwork with the highest association with survival. And then since we are considering the max statistic, essentially, we do a permutation test to make sure that what we see is not just the best p-value among many, and therefore is not just random noise. Okay. This is work done with Tommy Hansen, that was a master student at the University of Southern Denmark. Yeah?
So when it's done for case control studies or in general, what you're changing is the outcome. Instead of a survival time, you have a 0-1 outcome, or you have some, I don't know, some other type of outcome, but then the coloring mechanism and the search is the same.
They are very similar. The coloring mechanism is the same. You random color the vertices. The searching mechanism can change depending on the function that you are optimizing and what you are doing. It's used also for counting the number of occurrences of specific subgraphs. So it's not exactly the same, and there are different twists, but the basic idea is very similar. Okay. Okay, so now going back to the question of do we need to assume anything about the function? Well, as I said, color coding works very well, but what you need to assume is that your function is additive in the genes or in the sets. Okay? And the interesting part is that our score is not additive. And actually, Tommy Hansen spent, I guess, the first week or the first couple of weeks showing many instances where it was not, it wouldn't work well. So just, I guess, maybe he was a bit annoyed, and he kept coming to me and say, okay, in this way, that for these instances it doesn't work, for these instances it doesn't work, for these instances it doesn't work. I said, okay, that's great. Let's try to understand in which instances it may work. And so we have a theoretical result that shows that if essentially you have a subgraph associated with survival and the other mutations are placed independently, if you have enough samples, so if the number of samples is of this order, then using our approach, you will identify the subgraph associated with survival. So in the worst case, it will not find the subgraph with the highest association with survival. But if the data comes from the model that we think may be reasonable, then it should work well. Okay? Okay, so since I'm running out of time, I'm just going to mention something about we ran our algorithm simulated data. And the main point here is that for data sets of the size that we have now, they may not be large enough to identify subnetworks associated with survival reliably. Okay? So you don't always find the subnetwork really associated with survival, but quite often you find a good chunk of it. Yes?
Should we assume a K?
You were assuming a K, so we fixed, I guess, for these simulations. I don't remember exactly the K, but I think it was K equal to 5 or 6 in these simulations. But then what you can do is you can run your algorithm for different values of K and see if the p-values increases or not. So, but for these simulations, yes, we were assuming a K. Okay. And then we run it on some cancer data. I'm going to show you first the runtime. So our NOMAS is faster than the exhaustive algorithm, and luckily it is the case. Otherwise, there was no point in everything we've done. And then we compared our algorithm NOMAS with other approaches that are used in some studies to find subnetworks associated with survival. In particular, we compared with 3 different variants of greedy algorithms, where you start from one node in the network and you add the node that improves the log rank statistic the most, and you keep going until you reach a certain size or until the statistic doesn't improve anymore. And we also compare it with an approach in which you consider the function to be additive. So for example, very often people take the log of the p-values of the single genes, and now you try to find a subnetwork that maximizes the sum of the log of the p-values. And what the results show here for 3 different cancer studies is that while NOMAS always find the best a subnetwork with the highest association with survival compared to the other methods, sometimes the methods find results that are similar, but sometimes the results are very different. Okay? So using these heuristic methods that have no guarantees may not be always a good idea. Okay, so I guess I'm going to skip this slide since I'm already out of time. I'm just going to show you one result that is actually work in progress. So if we use it now, NOMAS for the same dataset that I mentioned earlier on glioblastoma multiforme, for k equal 8, this is the subnetwork that is identified. Okay? So the p-value for the subnetwork is very good, but maybe that's expected since we look for the best subnetwork. And then from the permutational p-value, what we get is around 0.1. So it's not great, not a great p-value, but it's much better than what you could do with the other methods. And in particular, no gene has significant association with survival when considered in isolation. So none of these genes is picked up by methods that look at one gene at a time. And also, maybe more interestingly, actually this subnetwork is part of 2 different pathways that were manually annotated in the GBM paper from Cancer Genome Atlas. And so this may be something that has also a biological meaning, but that's, of course, a big leap in reasoning. Okay, so I guess I'm already out of time. So I want to just thank the co-authors for the different works, Alexandra Papustaki, Ben Raffaele Eliupfal, that were all at Brown University at the time, Tommy Amundsen, that was at University of Southern Denmark, and this work has been funded by NSF, and University of Padova, and University of Southern Denmark. So if you have more questions, I would be happy to answer. I guess you were first, yeah?
Have you done like a power analysis to ask what kinds of effect sizes you'll be powered to use?
Yes, yeah, we have done some power analysis to see which effect size we need. And I can tell you more about that, but for the analysis that we have done, I guess we assume that the survival distributions for the mean of the survival time when you had the mutation was like half of the mean survival time of without a mutation in the network. So that was one example, but we have done more, and I would be happy to tell you more about that. Yes?
Yes, so you showed those QQ plots of the inflation, and then you developed this exact test to give better calibrated p-values from a log rank test. Did you try just like Bartlett corrections or other kind of higher order asymptotic things?
Yeah, so we tried some of the corrections that are reported in the literature for survival analysis. Some of them work well for some cases, but not all of the possible cases. So in particular, for us, we didn't see any result that gives you a guarantee that if you use a correction, then your p-value will be conservative, for example, and close enough.
The guarantee you have, I think, is novel. But I think some of those other methods would generalize to proportional hazard models, so you can bring in your covariates and pieces.
Yes, yeah. So an advantage of the other methods is that you can consider the other covariates. That's for sure. I guess we were looking more at this fact of having all these false discoveries. That's what really hit us. And I guess I didn't mention it, but an open direction is how to incorporate all the covariates in our framework that is not clear how to do it. At the moment, we just get the results with the method, and then we see if after correction for covariates, there is still significance or not. But of course, it would be interesting to put that in the computational problem. I guess, yeah. I guess there was one here, but
I was just wondering, you use the union now. Would it make sense to think about other not intersection, but other combinatorial
functions of the single genes? Yeah, I think so. I think it would make a lot of sense to consider, for example, the intersection. So if you see more than one mutation, that may be interesting. The union was based on this idea that you may have substituting genes with substituting functions. So they correspond to the same overall function, but mutation in two genes give you the same result. So that's why we use the union, but I think it's not the only one that one should focus on. No.
So, ignorant question. This gives you the best network, but you're making many tries with many random colors. What if you got more than one? I mean, and I don't mean this is a negative, it's a positive, right? Identifying not just the one that scores the best in this mechanism, but the top blah blah many.
Mm-hmm.
There may be several that are actually surviving. So could it be used this way? And also, this is really interesting. This coloring mechanism, it's a tool to parse and reduce the number of things that you look at every time you run.
Mm-hmm.
You do it randomly. Has anyone ever tried to do it not completely random, to sort of drive the coloring, so better exploit the
yeah, so I guess for the first part of the question, I guess, yeah, you could use, you get many solutions, you get tons of different subgraphs, and you could look at more than just one. Then I guess you can do a permutation test based on what you do to get a p-value, and we haven't done it. We look usually what we do is we get the best subnetwork, we take it out, and then we run the method again to see if there is something more. We often don't see more than one subnetwork. But maybe just because of the sample sizes, or maybe our method is not good enough. So that's for the first part. For the second one, I'm sure that in specific applications, people have looked at ways to not start, for example, from all possible genes, but to consider specific initial seeds for your solution. But at the moment, I don't have any of them in mind, if not for what we are working on. That is, if you have observed a subnetwork that has a given association with survival, and now you want to understand if in random data there is one that has an association at least as large as the one, then at least one of the genes should have not a significant association, but association at least as large as something. So it doesn't make sense to consider all possible subgraphs, but start from one of them and color the rest. And that's what we're working on. But I'm sure it has been, I believe it has been used also in other applications, but I don't have any specific one in mind. Thank you. Okay, I guess we're ready for the special coffee break.